Hi students, I am inviting you once again for the lecture. Now we are talking about the lesson Applications of Biotechnology. Under this we have studied 3 applications so far. 1. Medicine. 2. Gene therapy. The third one, what we have, stem cell therapy. And the fourth one, molecular diagnosis. Under the molecular diagnosis, we have discussed already the ELISA, enzyme-linked sorbent assay. This is one of the technique adapted in molecular diagnosis. Now the second technique adapted under molecular diagnosis is polymerase chain reaction. So it is nothing but an amplification process. If you want to get actually billions of copies of genes, that is being produced with the help of this reaction PCR, polymerase chain reaction. That's why it's called as actually the process called DNA amplification process. What is the meaning of PCR? So it is being done outside the body, not inside the body. Hence called in vitro amplification. So PCR is in vitro, that is outside the body. Amplification just to increase number or production of many copies, that is called amplification. An in vitro amplification technique. So used for synthesizing multiple identical copies of DNA of interest. DNA of interest means nothing but the known DNA what we needed. Suppose for example the DNA responsible for the synthesis of insulin hormone or growth hormone, they are called as DNA of interest or gene of interest. The desired DNA is called what is known as DNA of interest. So by this method we can produce multiple identical copies, not 1 or 2 but in billions of DNA of interest. So that's why it's called as gene amplification process or DNA amplification process. Generally we can say DNA amplification process. A segment of DNA is nothing but a gene, you know that one. That's why also I use the term amplification of genes. So the method was first developed by Polymerase, that is in 1983. He was awarded Nobel Prize in 1993. That's about what is called polymerase chain reaction, in vitro amplification of DNA of interest. Now during that process, we have normally 3 steps involved. 1. Denaturation. The second one, renaturation or primer annealing process. Then synthesis or primer extension. These are the 3 steps actually you have during the polymerase chain reaction. Now let's take about one by one. What is denaturation, renaturation, and also synthesis or primer extension. Increase in length, that is called extension of the DNA molecule. So all being done outside the body in a medium. So the medium contains the raw materials like the various nucleotides. Then we have one primer, then one DNA, then the enzyme, all being added in the form of a mixture to a medium, and where you can synthesize this DNA in billion copies. Just actually repeating the process, producing actually the DNA in billion copies. That's why it's called in vitro amplification. Outside the body, we are amplifying the DNA. There are 3 steps in PCR, polymerase chain reaction. Step number 1 I mentioned denaturation. Step number 2, annealing process. Annealing means sudden cooling, increasing the temperature to a maximum level and sudden cooling, and that is called annealing process. The third one, extension. Okay, now what is happening during denaturation? The double-stranded DNA of interest is denatured by high temperature. When exposed to high temperature, it is being separated. Denaturation is nothing but the separation of the 2 strands of DNA to form single stranded structure. That is being done with the help of temperatures, an example of 95 degrees Celsius for about 1 minute, so that we can separate the 2 strands. That process is called denaturation. So to separate 2 individual strands by high temperature, this is called denaturation process. Once we are getting the single stranded DNA, the single stranded DNA is allowed to hybridize with what is called a primer. You know, in the case of DNA replication, the DNA polymerase cannot initiate the process. A small segment of DNA is normally, or RNA is added, a small segment of RNA is added as a primer, and that is the one which initiates the replication process, and that is what we call the primer. So the single stranded DNA is allowed to hybridize, means there is actually pairing, and that is called hybridization. This process is called renaturation or primer annealing. So here increase to temperature, here sudden cooling, and then hybridization process between the single stranded DNA template and the primer. The third one, once actually the primer is hybridized with a single stranded DNA, the next step, the primer template is used to synthesize DNA using what is called one enzyme that is specific for PCR, that is called Taq DNA polymerase. Now this is the enzyme, a thermostable enzyme. This is a thermostable enzyme. The meaning for that one, it can withstand high temperature. Normally the enzymes are protein in nature, they will be denatured after even 40 degrees Celsius. But this is an enzyme which can withstand, you know that one, a high temperature of more than 95 degrees Celsius. That's why it's called thermostable enzyme, thermostable DNA polymerase. Now it is being obtained from a bacterium, an archibacterium, Thermus aquaticus. Thermus aquaticus. This is the name of the bacterium from which what we call the enzyme DNA polymerase is obtained. So this is what's called thermophilus bacterium, the one which can live in extreme conditions, extremophilus bacteria. So there you have Thermus aquaticus, this is the bacterium from where the enzyme is obtained, hence called Taq DNA polymerase. It is a thermostable enzyme, the meaning it can withstand a high temperature. Now we'll see these three steps in detail, okay, along with the diagrams. The steps involved in PCR, there are three steps I mentioned. Number one, denaturation. So during denaturation, what you have to do, the reaction mixture. The reaction mixture is normally taken in what we have in a glass vial or petri dish. Just we have a vessel consisting of primer, nucleotides, the raw materials, the Taq DNA polymerase, and a double-stranded DNA which is acting as a template. That is known as the reaction mixture. So consisting of primer, nucleotides, Taq DNA polymerase, and double-stranded DNA which is acting as a template, or the DNA of interest which is to be multiplied. So the DNA, double-stranded DNA is nothing but the DNA of interest which is to be actually multiplied. So the mixture is heated to just 95 degrees Celsius for a short time. The book is given short time, but the duration is about 60 seconds or 1 minute to denature the target DNA. The DNA which is to be amplified is called as a target DNA, the one which is to be amplified, into single strands that will act as templates for DNA synthesis. The two strands get separated, as in the case of replication process. Each strand is capable of acting as a template to synthesize its own complementary strand. That is what is called denaturation, separation of the two strands with the help of high temperature of 95 degrees Celsius for about 60 seconds. It will not be extended beyond that level. Then the next one, primer annealing. Annealing means nothing but actually sudden cooling process. That may result sometimes in melting. Normally we can say melting. So annealing is done by rapid cooling of the mixture. So from 95 degrees Celsius to 54 degrees Celsius, that is retained in the temperature for about 45 seconds, a short duration. So rapid cooling of the mixture and allowing the primer to bind to the sequence of each of the two strands. In each of the two strands, we have the two strands are separated, they have the base sequences. Now this primer is normally binding to the sequences on what we call the template DNA strand, which is of interest. To two strands of DNA flanking the target DNA. So they are flanking just actually on either side. The meaning for flanking on either side or simply just attached to. So that's about the primer annealing process. Here the annealing is nothing but sudden cooling and hybridization of the primer with what is called the single-stranded template DNA. Don't forget the reaction mixture is the one which consists of the primer to initiate the process, the nucleotides, the raw materials, then the enzyme tagged DNA polymerase for what is called extension, and a double-stranded DNA which is acting as a template as in the case of replication process to produce new copies of DNA of the same type. That is called DNA of interest. Now the third step in what is called PCR reaction is synthesis of DNA or extension of what is called primer DNA. Now during synthesis what is happening? The DNA extension, the temperature of the reaction mixture. I mentioned reaction mixture is nothing but a combination of DNA template, then we have the enzyme tagged DNA polymerase, the raw materials, the nucleotides, okay, and also we have a primer DNA. So what is happening? The mixture is heated to 75 degrees Celsius. The first time 95, then sudden cooling to what is called 54 degrees Celsius. Once again we are increasing the temperature to 75 degrees Celsius for a sufficient period of time. The time is about 2 minutes. What for? To allow the tagged DNA polymerase to extend the primer. Now what is happening? To extend the primer is being extended by copying what is called the template single-stranded DNA. By copying the single-stranded DNA template. The two strands separated, after that each strand is acting as a template. By using the template, the extension of the primer DNA is taking place with the help of the enzyme by adding the nucleotides. We'll see the picture. So at the end of incubation, both the single template strands made partially into double-stranded. The process is going on, double-stranded DNA. So the new strand of each of the double-stranded DNA extends. So one old strand, another one is a new strand, that is nothing but the extended primer DNA. Extends to variable distance downstream, that is from 5' to 3' or 3' to 5'. This is called what's called downstream. Either from 5' end to 3' end or from 3' end to 5' end according to orientation. So the process is being repeated again and again to generate billions of copies, multiple copies of what is called the DNA, that to the identical form of DNA. That's why this process is also called DNA amplification, production of billions of copies of DNA, hence called actually DNA amplification. Now I'll show the picture, now we can understand, a very simple picture as given in the book. Okay, this is an illustration showing what are the steps involved in PCR reaction, what I described. Number one, DNA primer, I made a mistake. Number one, denaturation process. Now we have a mixer that is in a vial or in a petri dish or in a glass plate. The mixer consists of, I mentioned about the mixer consists of a DNA which is acting as a template, then the raw materials, nucleotides for the formation of a DNA strand, then the original DNA, that's a template. We have DNA primer. This primer is nothing but a small chemically synthesized oligonucleotide complementary to the region of DNA. Small, just actually chemically synthesized oligonucleotide. Oligo means made up of only a few nucleotides of about nearly 11 to 12. Now the mixture also contains what is called Taq polymerase enzyme. Taq DNA polymerase also present in that mixture. This is the one responsible for the synthesis. Thermostable enzyme which can resist temperature even above 90 degrees Celsius. That's why it's called thermostable enzyme. Now this is a mixture. The mixture is allowed to undergo denaturation by increasing the temperature of 95 degrees Celsius for about 1 minute. So the two strands get separated. So the opposite strands get separated. The next one, annealing process. This is nothing but the sudden cooling to 54 degrees Celsius for about 45 seconds. So that what is happening, the primer hybridized with what we have, the single strand of DNA. Now this is a DNA primer. Here also DNA primer. So the base pairing between this DNA primer and the single stranded DNA is called as hybridization. So DNA primer hybridized with the single stranded DNA, that is nothing but the pairing process. Now we have the extension of the primer to form the entire length of variable distances according to the length of the DNA what we have given, the DNA of interest. Now the process is being actually carried out by the enzyme DNA polymerase. You know the synthesis always occurs in 5' 3' direction as in the case of replication. In one case it is from 5' to 3' in this direction. In another case it is in this direction from 5' to 3'. Anyway, in both the cases the synthesis, the extension of the primer occurs only in 5' 3' direction. Now within 30 cycles the process is being repeated. I mentioned 1 billion genes or 1 billion DNA copies are made. In 30 cycles, so the process being repeated again and again. In 30 cycles, after the 30 cycles, you can get 1 billion DNA copies. So that's about what we got the steps in PCR reaction. Okay, they separate the strands, then what is called the primer annealing, cooling down, and then hybridization, together calls annealing process. Two events happening, cooling and then hybridization. And here only the elongation of what is called the new strand. So here the new strand is complementary to what we have, that is a original strand. I mentioned already the DNA primer is a small segment, a chemically synthesized oligonucleotide having 10 to 12 nucleotides, hence called oligonucleotide. The bases in this one are complementary to the bases of DNA. This is nothing but the DNA primer. Okay, that's a step. Now we have to go for one more. So this technique can also be used for the amplification of RNA. What is reverse transcription PCR, polymerase chain reaction, or RT-PCR? Now normally we are using this actually technique for the synthesis of, for the amplification of DNA. The same technique can also be used for the amplification of RNA. How? Normally in the case of RNA, if you want to analyze the nature of the protein in a cell, you have to probe what is called mRNA. Because normally the mRNA carries the codon. The bases in mRNA are complementary to what is called the DNA, one of the strands of DNA which is acting as a template. Now when mRNA undergoes the reverse process, normally DNA directs the synthesis of RNA with the help of DNA polymerase enzyme. Here what is happening, mRNA directs the synthesis of DNA. And that DNA is called complementary DNA by a process of reverse transcription with the help of reverse transcriptase enzyme. This reverse transcriptase enzyme is nothing but DNA polymerase, RNA dependent DNA polymerase, working under the control of RNA. This is called RNA dependent DNA polymerase. So in the case of cDNA, it's called complementary DNA because the bases in DNA are complementary to the bases of mRNA. So it is nothing but copies of mRNA. So from this single stranded cDNA, we can synthesize another strand of DNA by means of polymerase chain reaction using primer. Here we are getting only one strand because mRNA is a single strand structure. So we are getting what's called single stranded DNA, that is complementary DNA, which is normally complementary to the basis of mRNA. And from this single strand we can produce what we have double stranded DNA by using polymerase chain reaction, after which we can produce many copies of DNA. Okay, Now I mentioned about PCR technique can also be used for the amplification of RNA, in which case it is referred as reverse transcription polymerase chain reaction or simply RT-PCR. Because we are producing the DNA, then amplifying the DNA based on reverse transcription method using the enzyme. So in this process, RNA, particularly the mRNA molecule, what I described here, must be converted into cDNA called complementary DNA by the enzyme reverse transcriptase. Now the cDNA then serves as a template for PCR. The DNA has been used as a template for the synthesis of a double-stranded DNA and many copies of DNA. That's why it's called as actually cDNA, complementary to the basis of mRNA. So that's all about the mechanism related to the polymerase chain reaction. We have to go for what is called the role of PCR, particularly in diagnosis of disease, and also the applications. We'll see in the next class.